As we begin to study skeletal muscle, there are a few concepts we need to cover. Skeletal muscles can be classified according to how they function. Prime movers are muscles that are primarily responsible for the movement. Antagonists are muscles that oppose the prime mover. Synergists are assistant muscles. They help assist the prime mover either in making the movement, or many of them act as fixators. They help stabilize the bones for the prime mover to be more effective. Muscles have to cross joints and pull on bones for movement to occur. The origin of a muscle is the attachment of the muscle to the bone that does not move when the muscle contracts. The insertion is the attachment of the muscle to the bone that does move when the muscle contracts. There is quite a bit of connective tissue in muscles. The muscle attachments are made of connective tissue. If the attachment is a rope-like structure of connective tissue, it's known as a tendon. But flat muscles tend to have a sheet of connective tissue come off the edge of the muscle that attaches this to the bone or to the surrounding structures. This sheet-like structure is known as an aponeurosis. Muscles have a number of connective tissue coverings. The fascia is connective tissue that bundles muscle groups together, things like the quadricep. The four muscles that make up that group of muscles are bound by fascia. Each individual muscle is covered with an epimysium, a connective tissue that's known as the epimysium. Muscle fibers within a muscle are arranged in bundles known as fascicles. Each fascicle is surrounded by a connective tissue sheet known as the perimysium. And each individual muscle fiber or muscle cell is surrounded by connective tissue known as the endomysium. So as we look at the muscle, here we see that rope-like tendon that's attaching the muscle to the bone. We don't see fascia here because we're only looking at a single muscle, but here is the epimysium that surrounds the entire muscle. Notice how the muscle is made up of bundles of muscle cells that are the fascicles, and the fascicles are surrounded by the perimysium. And then each individual muscle cell is surrounded by its own connective tissue sheet known as the endomysium. As we look at the skeletal muscle anatomy under the microscope, we're going to notice that the muscle is striated. Sarcomeres are the contractile unit of the muscle cell, and there are thousands of sarcomeres in each muscle cell. Myofilaments are organelles in the muscle fiber or the muscle cell that are filled with cytoskeletal elements. These are the proteins myosin and actin. Myosin and actin are overlapping, and this is what gives the cells that striated or striped look. Myosin and actin slide across each other during muscle contraction, and this is what leads to the sliding filament theory for muscle contraction. So here we see a part of a muscle cell, a myofibril out of a muscle cell, and a sarcomere runs from Z disc to Z disc. Now the actin filaments, the thin filaments, are attached to each Z disc. The myosin filaments, or the thick filaments, are held here at the M line. You can see how actin and myosin overlap, and this is going to give us our striations. Thin filaments only, thin and thick filaments only. When actin slides across the myosin, it brings the Z discs closer together, shortening the sarcomere. This happens in every sarcomere up and down the muscle fibril, and this is what causes the muscle to shorten. There are three types of muscle tissue. Skeletal muscle is attached to bones. Smooth muscle is found in the walls of hollow organs and tubes, and cardiac muscle is found in the wall of the heart. Skeletal muscle is that striated muscle. You see the stripes, that's the result of the overlapping myofilaments. These cells are extremely long. They go the entire length of the muscle, and they're multinucleate. We say that this is striated voluntary muscle because we consciously control the contraction of skeletal muscle. Here is smooth muscle. Here the cells are spindle-shaped, and they tend to arrange themselves in sheets. Smooth muscle is non-striated. There's a different arrangement of myosin and actin filaments, and this is involuntary muscle. We do not control when this contracts. And then we have cardiac muscle. Cardiac muscle is striated. It has the same arrangement of myosin and actin as we see in skeletal muscles, but the cells are branching. As a result, the cells have to be connected to each other, and that's what these intercalated discs are. So cardiac muscle is striated because of the stripes, but it is involuntary because we do not consciously control when it contracts. There are several criteria that are used when we are naming muscles. One is the direction of the fiber. If rectus is in the muscle name, it means the fibers run straight up and down. Oblique would mean the fibers run diagonally. Another is muscle size. We talk about minimus, medius, or maximus muscles, indicating that they're small, medium-sized, or large. The muscle location is always helpful. If there is something in the name that tells you where the muscle is, like brachialis, you would figure that one's in the arm. Femoris, that would be in the thigh somewhere. Also, we can use the number of origins or heads. Biceps would have 2 origins, triceps would have 3 origins. The origin and insertion of the muscle may be used as part of its name. The sternocleidomastoid is a muscle that originates on the sternum and the clavicle, cleido is clavicle, and inserts on the mastoid process of the temporal bone. Sometimes the shape of the muscle is used. Deltoid is a triangular shaped muscle. Trapezius is sort of a kite-shaped muscle. And sometimes we use the action of the muscle, flexor, extensor, abductor, or adductor. If you learn to look at the names and what the naming criteria is, a lot of times the name of the muscle will tell you a lot about it.